Hello, this is Dr. Jabbour, and today we're going to talk about cellular respiration. So, what is cellular respiration? So, as the definition here states, it's a set of metabolic reactions that take place in your body with the purpose of breaking down the food that you're eating to produce energy in the form of ATP. So the food you ingest, the purpose of it is to bring you the fuel that your body needs to do the things that your body does. So the food has energy, and cellular respiration is about taking that energy out of the food and distributing it to the cells. So this is the chemical reaction, like the overall chemical reaction. There are actually several phases in cellular respiration, and we're literally going to go into each one of these phases and look at the nitty-gritty and really the details of it. So this is just kind of an overall view of what's going on overall. So you ingest food, and we're talking here about sugar, glucose. This is a monosaccharide made out of 6 carbons, C6H12O6. And you're going to process that glucose for the purpose of producing energy, which fuels your body in the form of ATP. In the previous lecture, we talked about ATP, how ATP, this one small molecule, has so much energy in its bond between its second and third phosphate, because ATP stands for adenosine triphosphate. So to make ATP, we need glucose, but we also need oxygen. This is considered to be an aerobic reaction. So aerobic respiration uses oxygen to do the process. And you'll see where oxygen comes into the story. It actually comes all the way at the end of the story. So at the end of this lecture, we'll say, hey, this is where oxygen comes into the story, and you'll see what it does. It's very cool. So oxygen will actually come at the end. The first phases of cellular respiration deal with glucose and how we have to break it down first and create some intermediary molecules that are going to be very important for the last phase of cellular respiration. In the process, you generate byproducts, and one of the byproducts is carbon dioxide. So this is a gas, just like oxygen was a gas. And by the way, in chemistry, this little arrow pointing upwards is representative of a gas. So oxygen is a gas you breathe in, you need it for cellular respiration. Carbon dioxide is a gas you exhale. That's a byproduct of cellular respiration. So it's a waste product. You don't keep CO2 in your body. Excuse me. Another product of cellular respiration is water. So in the process of doing cellular respiration, you also release water molecules. Okay, so overall, this chemical reaction will end up releasing as many as 30 to 32 ATP molecules for every 1 glucose. So if we have 1 glucose here, we end up with around 30 to 32 adenosine triphosphate molecules. Okay, so that's the big picture. You absolutely have to know this. You have to know that there's glucose and oxygen as your substrates, your reactants, and the products at the end are carbon dioxide and water, and of course the energy which is the purpose of cellular respiration. Now, one more thing before I move on to the next slide. I want you to notice that every single one of these atoms is accounted for on the other side of the equation. We might not expect you to, know all the chemical reactions and the details of that, but for the overall reaction, it's kind of important for you to see that we start here with 1 molecule of glucose that happens to have 6 carbons in it. And these 6 carbons, we actually are going to find them in the CO2 molecules. One CO2 molecule only has one carbon. So really, in the end, you're going to exhale 6 carbon dioxide molecules. Carbon dioxide are very small molecules that you exhale, made out of 1 carbon and 2 oxygens. This is CO2. Well, 1 glucose molecule, because it has 6 carbons, is actually going to generate 6 molecules of CO2. So you see this number 6 here in ahead of CO2. That's to represent how many CO2 molecules we actually are generating. So if you notice, every single one of these atoms is actually accounted for on the other side of the equation. All right. So that was the big picture, from glucose and oxygen all the way to energy, with a couple of things that are going to be produced, which are carbon dioxide and water. But what are the actual stages of cellular respiration? Well, there are 3 main stages: glycolysis, pyruvate oxidation, and the citric acid cycle. And the last and final stage is the oxidative phosphorylation, which includes electron transport and chemiosmosis at the membrane of the mitochondrion. Okay. So I'm planning to draw you, another kind of like big picture of, okay, where's this food coming in? Where is it going? Where is cellular respiration really taking place? Because we haven't said that yet. We haven't talked about that. We just talked about the chemical reaction of what comes in and what comes out. So let's look at this little emoji here. So this is just to illustrate what's going on. You're going to eat food, and let's assume it's a cake because we're talking about sugar. So here we go. You ingest the cake, and it's full of sugar. And here's your esophagus and your stomach. And so the food's going to go down your stomach, and what's around your stomach, and this is very exaggerated, are blood vessels. So this is blood. Fine. Your food, so the cake I drew it pink, so I'm going to do little pink arrows here. The food's going down the esophagus, in the stomach. Really, let's be a little bit more accurate than that. You know what? There's no room to draw intestines, so I'm just going to draw one long tube without illustrating the stomach. So normally there's a pouch called the stomach, and then it goes into the intestines. So there's really no stomach here, but that's okay. All right, so the food eventually it ends up in the intestines. And it's in the intestines, my GI is not very accurate here, but it's in the intestines that the food is going to get absorbed and where? Into the blood. That's the purpose. We eat, our intestinal cells break down the food, and these tiny little sugar molecules get absorbed literally through the intestinal wall and into the blood that's right behind the intestinal wall. So the food is literally going from one cavity into a cell, into the blood. So you end up with glucose right here. Glucose right here. The glucose is going to be taken up by all the cells in the body. So this blood is delivering glucose to every single cell in your body. It's literally like, here's some glucose, here's some glucose, here's some glucose. It's literally like going, flowing through all of your vessels and distributing the glucose to your cells. Now, in a cell, so this is a cell right here. Right here. So this is one of the cells that's going to receive the glucose. It receives the glucose by way of a transporters, because the glucose doesn't enter the cell just like that. So it leaves the blood and enters the cell via transporter, a glucose transporter. So when it enters the cell, this is when those three phases start. So that's why I wanted to draw this, to give you the context, the anatomical context of where this is happening, not just the chemical reaction of we start here, we get this, but where is this happening? It's happening in each individual cell. Every cell in your body is doing that. It's receiving glucose from the blood, and it's also receiving oxygen, uh, right? And you know, if you cut off the circulation to, for example, a finger, it can die off because all the skin cells are going to die because they're not getting blood, which provides the oxygen to let the cell survive. So this blood is providing oxygen, but also glucose. So glucose enters the cells, and then that's when the three phases start. It's in the cell. So the first phase is glucose turns into pyruvate. This is phase 1, which we call glycolysis. So I want to go back here. This is it. I'm talking about these three phases or stages. One is glycolysis. Okay. The second one is pyruvate oxidation and the citric acid cycle. So pyruvate oxidation takes place still here in the cytosol of the cell. But then it's going to enter the mitochondrion. And this is very exaggerated because a mitochondrion that big in a cell, that's impossible. So this is a mitochondrion with this double membrane, right? So this is an organelle inside the cell. The pyruvate is going to enter the mitochondrion, and there will be this the second phase with the citric acid cycle. So I'm just going to draw a cycle like that, like this circle, just so a cycle. So I'm just going to put 2 here. We'll get into those details, you know, when we actually see each one of them. I just want you to show the big picture. And then the third phase, the chemiosmosis, oxidative phosphorylation, which is electron transport and chemiosmosis, is going to happen at the membrane, the inner membrane of the mitochondrion. Okay? So all of this is happening inside the cell, in very specific parts of the cell, different compartments of the cell. When we talk about each one of them, I'll specify where it's taking place. But this is where glucose is coming from. It's coming from the sugar that's inside the cake that you just had a bite of. And that sugar, well, the cake is going to get broken down into, you know, smaller molecules. And then finally, the tiny sugar molecules will be absorbed through the intestinal wall into the blood and distributed to all the cells. And after glucose enters the cells, then this process starts with the stages 1, 2, and 3. Okay. So in a textbook, this would be the diagram that's kind of provided in a textbook with a glycolysis here outside of the mitochondrion. So here we're assuming this tan background is the cytosol, which is the liquid environment that's inside the cell. It's written here, cytosol, right here. Right. So this is the cytosol, and then there's here a mitochondrion that's illustrated. There's the mitochondrion, and that's one of the organelles the cell has. That's the organelle we're talking about today because that's where the magic happens. Okay. So glycolysis takes place in the cytosol. Pyruvate oxidation and citric acid cycle takes place in the mitochondrion. Oxidative phosphorylation takes place in the mitochondrion's inner membrane, even more specific. Okay, so this was stage 1. This is stage 2. And actually, I'm going to move 2 over here because it includes both of these, pyruvate oxidation and citric acid cycle. And then this is stage 3. Okay, right? You see the big picture? Okay. Okay. So the goal is to make ATP, and most of the ATP is made at the end during the oxidative phosphorylation. However, there's a little bit of ATP made during glycolysis, just 2, and there's a bit more ATP made during the citric acid cycle. The majority of the ATP will be made all the way at the end. Let's talk about this. Okay. So a very important concept to understand even before we get into the nitty-gritty is the concept of electron transfer. This is really how it's going to work. Electrons are going to get exchanged from one molecule to the next molecule to the next molecule to the next molecule. And there's going to be release of energy when that's happening. The rearrangement of these electrons is the source of that release of energy. And that release of energy is harvested, is taken into making an ATP molecule. So it's important to understand, what is an electron transfer? It's basically breaking bonds and reforming bonds, because that's what an electron is. It's about an electron that's going from one molecule to another molecule, and electrons are involved in bonds. That's how they are. And during that electron transfer, but from one molecule to the next, there's actually a release of energy every time. Remember, that's the energy that's going to eventually be used to make the ATP. It's hard, right, to think about. So we're releasing energy to make energy. Yes, we're releasing energy from this transfer of electron to make this bigger energy that is basically all included into one molecule that we can distribute to the cell and can be used for, different jobs within the cell, like the protein pump and the membrane. Okay, so we're going to see these electrons move down the electron transport chain in that last phase of cellular respiration. And these electrons we're going to see are releasing, the movement of these electrons are releasing the energy that's going to be harvested and put into ATP molecules. So as I told you, oxygen is going to be needed. It's an aerobic chemical reaction. Oxygen will be needed because oxygen is the last electron acceptor. So there's a chain here of events happening, one molecule giving off the electron to another molecule, etc. But the last molecule, what is it going to give its electrons to? So it actually gives it to oxygen. Excuse me. And oxygen turns into water, and that's okay. We basically took these electrons and buried them in water, and now we don't have to transfer them anymore. And we're at the end of the chemical reactions anyway. We made our ATP, everything's good. We use the electrons as the last, we use the oxygen as the last electron acceptor of this chain of events. So we're going to get back to that when we get to that last phase. So aerobic respiration, it ends up being a series of oxidation reduction chemical events that are also called redox reactions. In redox reactions, the electrons are lost by one substance, but they must be picked up by another substance, right? It's like released and accepted by another one, released and accepted by another one. And the last one that accepts the electrons in this whole story is the oxygen. So there are a couple of examples on the upcoming slides that are not directly related to cellular respiration, but just to show you with atoms, because you should be familiar about what, about atoms and how they lose electrons and gain electrons, because that's something we discussed in the chemistry, the basic chemistry lecture that I had for, biology. So the definition of oxidation is loss of electrons. Loss of electrons, and that electron will go to another substance. So the substance that loses the electron, it becomes oxidized. If you want to remember, oxidation is losing an electron versus reduction is actually gaining an electron. I'm going to give you a trick. Loss has an O just like oxidation. You have to find a trick because, well, maybe you're not going to be like me, but as a student, I could never remember which one's which. Oxidation. Do we lose an electron? Do we gain an electron at oxidation? I could never remember it. But I'm giving you a trick here. Oxidation is the loss of electrons, okay? And there's an O in loss, like oxidation. All right. In a reduction reaction, it's an addition of an electron on the substance. The substance that gains an electron becomes reduced. So if you remember O for oxidation, so loss, you remember reduction is the other one. So here is an example here, sodium chloride and sodium chloride. So we saw back when we did some basic chemistry that sodium atoms, they need to get rid of an electron to be stable. Because if you remember, in its valence shell, there was one too many electrons. Because in the valence shell, it needs to be a number of 8 or a factor of 8. But in the valence shell needs to be full, or a number of 8. So if we have 9 electrons on the valence shell, on the last shell to be filled, that's one too many. So, we can bring in 7 more, and now we have another group of 8. But bringing 7 electrons from 7 other atoms, that's kind of too difficult. So it's easier to ditch that one electron that's too many and get rid of it. So sodium atom will ditch an electron and become a sodium ion. And this is an ion, or more specifically, a cation, because it's positive ion. Cation. Who's going to be happy about that is chloride. Yes. And I forgot if this is chloride and this is chlorine. I might mix these two up. Some things I don't, I still don't remember. Okay. So this one actually is 1 electron shy from being full on the valence shell and just requires 1 electron. So it has 7 electrons on its valence shell and really would like to have 1 more. So it's a factor of 8, right? It's 8 electrons on the valence shell, so it's going to be a very stable atom. So by gaining this electron over here, it has 1 too many electrons as far as if you're counting protons and electrons. So that's why there is a negative sign here, because we have more electrons than we have protons. Remember, protons and electrons in atoms, they're equal because atoms are overall neutral. When you get an atomic number of whatever it is, 11, then that means there are 11 protons, but that also means there are 11 electrons that are flying around the atom. And because atoms are overall electrically neutral. How many numbers of protons there are, that's how many numbers of electrons are gravitating around the nucleus. So here we have a gain of an electron. So we have one more electron than we have protons. That's what the negative sign is showing you. And here we have one less electron than protons for that in that ion. So we put a plus sign here. Okay, and they both become satisfied. They're both satisfied because their valence shells are full. So we say that sodium atom becomes oxidized into the sodium ion, and chloride atom becomes reduced into chlorine. Okay, so, and look at this. This is exactly how electrons get transferred. They are transferred from A to B. So we had sodium, and sodium lost its electron and gave it to chloride. And now we have a new molecule with these electrons kind of added to it. So this is the case for many redox reactions. Well, all of them, because that's what the definition of a redox is. There will always be one substrate that is going to be reduced because it's going to be gaining electrons. And you can see that here it used to be a cation with 2+. That means it had it was missing 2 electrons. So so to get reduced, it has to gain these 2 electrons. So when it gains the electrons, it doesn't have this 2+ sign because it has these 2 electrons now. And these 2 electrons, where did they come from? They came from the other substrate that lost 2 electrons and became oxidized in the process. This is another example of redox. This is methane. Methane reacts with oxygen gas. And what happens when you have the 2 together? You can have carbon dioxide produced with energy release and water molecules, because we have to kind of take care of those atoms that are within these molecules. So the other atoms rearrange to become water molecules. So the carbon, there's only one. See, so if you only follow the carbon, this is it. This one carbon. Okay. This is the diagram that goes with each one of these molecules to show you where the electrons are. So methane has electrons at each, of course, at each of these covalent bonds. If you remember, electrons are shared, right? So there is an electron from hydrogen that is given to carbon, and there's an electron from carbon that's given to hydrogen to fulfill both of their needs. So hydrogen receives an electron, and a hydrogen which actually has an atomic number of 1, that means it only has 1 electron. And 1 electron on that first shell fills up at 2, not 8. The first shell is the exception, it's just 2, and then it's 8 and factors of 8. So the first shell is 2. That means if you have an atomic number of 1, you only have 1 electron. That's not stable, because 2 is stable. 2 or factors of 8. So we need an electron from somewhere, and this is the role of carbon, is to give an electron to hydrogen, so hydrogen can see both of these electrons for itself. There you go. So they're shared. This is what we say. They're shared. Okay, so it's perfect for hydrogen, but for carbon, we need 3 more partners because carbon needs 4 additional electron. Hydrogen only needed 1 additional electron, so it can just partner with 1 other atom and it's fine. It's fulfilled. Carbon needs 4 partners, so it will partner with 1 hydrogen, but that's not going to be enough. So methane happens to be the example where the other 3 partners are also hydrogen. So an oxygen molecule looks like this with 1 double bond, and then CO2 looks like that, and then the water molecule. Excuse me. All right, so here's the big picture again. All right. So we need to these electron transporters we've been talking about, and we gave the examples in those redox reactions as, oh, we talked about sodium that turns into sodium ions, and we talked about methane, and and they're going to release electrons and pass it on. Well, there are very specific molecules that are used in cellular respiration to transport these electrons and pass them on one to the other until you reach the end of that chemical reaction. These I called electron carriers. So again, I'm talking about molecules that will be transporting that electron from one molecule to the next. Yeah. So the first molecule is called nicotinamide adenine dinucleotide. If you don't remember the name, it's okay, but NAD you need to remember. So NAD is an electron carrier that's going to be used during glycolysis, but also during the citric acid cycle. It has an oxidized form called NAD+. Remember, O for oxidize is the same as loss. And look at this, it lost an electron. NAD+ lost an electron, right? NAD+. Now it has 1 more proton than it has electrons. When it gains an electron, it's NADH. Because now it has an electron, and oh my gosh, I have an electron, I can bind with a hydrogen. So NADH is the reduced form. So this is the reduced form. The oxidized form and the reduced form was written right here. Okay. So how does this work? Hydrogen from our food, this is a great example, is going to be used to reduce NAD+ or oxidate NAD+. So what does that mean? That means that the electrons that are coming in are going to be used to reduce NAD+ into NADH. Once I go through the process, you'll understand. And then we have a second electron carrier. It's called FAD. And FAD actually can bind 2 hydrogen, so it's FADH2. So both NAD and FAD are going to be used as electron carriers. They're going to carry the electron and pass it down, and it will be very important for that last step of cellular respiration. Okay, so just like NADH, FADH2 is going to be used as an electron transporter across that electron transport chain. So I wanted to just write it out just just one more time with NAD instead of just sodium or methane, right? These other examples that were written previously. I want to actually write the ones that are in the story. So I just wanted to make that clear. So I wrote here NAD plus in its oxidized form is going to become, is going to become NADH H plus. This is kind of how we actually write it. That's the reduced form. Okay, and how does that happen? That happens because we have the arrival of two hydrogen atoms. And this is coming from the food. So when you eat and the food is broken down, atoms are going to be released, and hydrogen is kind of part of that. This is how it's used. So two hydrogen atoms from the food are going to be used to basically reduce the oxidized form of NAD plus. Yeah, I'm just going to leave it at that. And same thing with FAD. FAD also needs two hydrogen atoms and will become FADH2. All right. And I want to write that down here for you with the glucose molecule. So where's this H coming from? I'm going to show you exactly where. So I'm going to write C6H12O6. That's glucose. Right? This is glucose with a 6-carbon molecule, carbon atom, one molecule that it contains 6 carbons in it. And with 6 oxygen molecules of gas. And we end up with 6 CO2 molecules of gas plus 6 water molecules. Remember, I told you CO2 and water are the products that are released by cellular respiration, plus, of course, the ATP, which is the whole purpose of doing all of this. Okay, so I wanted to show you that, well, remind you that those 6 carbons, this is where they are. 6 1-carbon CO2 is the same as 6 carbon into 1 molecules of glucose. So I just wanted to show that to you. And I want to show you where the H's are coming from. So let's, here, this H and this H, right? The H's that are going to end up here. These H's, they come from here. This is where they are. Right? This is where they come from. They come from the sugar. The hydrogen are going to be used for making the electron transporters. So, and here they are, right? So those 12 hydrogen, they're here. So 6 times 2 is equal to 12. So they're in the water molecule in the overall reaction, but in the process, they will actually go through NAD and FAD first. This is just all the way at the end, we end up with water molecules. Okay, so now that I kind of like explained those 2 electron acceptors and really the overall reaction one more time, now we can get into the story. Can you imagine? We're just starting now the story. All right. So the first part of the story is glycolysis. And remember, there are 3 stages, and the first stage is glycolysis. Okay. So glycolysis takes place in the cytosol of the cell. This is very important to know. Typical test questions. where does glycolysis take place? So you have to remember, it's not in the mitochondrion. We're just in the beginning of the big story. In the beginning, we're just in floating in the cell. And what makes us float in the cell? It's the cytosol. It's that liquid in the cell. Here's your glucose. 1, 2, 3, 4, 5, 6 carbons. All right, 6 carbon chain. Technically speaking, it's not linear. It's in a cyclic molecule, but that's okay. It's 6 carbons. What do we end up having at the end of glycolysis? We end up having 2 pyruvates. So you need to know the name of that molecule, and you need to know that there are 2 of them. Believe me, I'm going to show you all the things you don't need to know, and that's going to be kind of very too detailed for the purpose of this course, this lecture. But this is definitely the minimum. So a 6 carbon molecule becomes 2 3 carbon molecules. See, 1, 2, 3, 1, 2, 3. It's as if you took something to cut this molecule in half. That's exactly what you're doing. The glucose molecule is going to be split in half. And there are steps to this splitting, but this is the overall reaction. We're going to have the use of ATP in the beginning. It's like, we're trying to make ATP. Why are we using ATP? Because we need it. We need ATP in the beginning to split the glucose molecule. That's a reaction that requires energy. But then we have the creation of NAD plus, NADH, which is the reduction of NAD plus. NAD plus is the oxidized form, and NADH is the reduced form. We need NADH for future use in the story. So NADH is getting generated here for good reason. And in the process, we also have ATP production. Perfect. That's what we want to make anyway. This is the whole purpose of the big story, is to make ATP. So not that many. We make 4 ATP made. But really, we consume 2. So technically speaking, we only have 2 ATP really produced, because we used 2, we made 4. How many did we actually kind of, gain? And how many are we left with? It's just 2. So glycolysis itself, so this, you see just this one big long line, but it's actually many little lines. It's a series, it's a many enzyme catalyzed reactions. There are many little reactions that are taking place, one after the other, after the other, after the other. Every reaction has its own enzyme, very specific to that particular substrate. And now we just learned about enzymes in the lecture on enzymes, ATP and enzymes. And without these enzymes, we wouldn't be able to catalyze the reaction, and it would never happen. So glucose is partially oxidized and broken down into 2 smaller sugars called pyruvate. So here are the steps of glycolysis. And right away, you can see that, oh my goodness, that's a lot of molecular details that are not necessary to, to understand at this point. But you need to understand the overall outcome. You need to know the outcome of this. So this is the first, remember, stage, the glycolysis. So here's your molecule. This is your glucose right here. And this glucose has 6 carbons. So here's 1, 2, 3, 4, 5, and the 6th one is up in the air. So there are 5 carbons that are cyclic and producing this this monosaccharide in a cyclic form, right? They're all linked like this, and it's forms a cyclic molecule. Well, those are 6 carbons. So what do? We need to change the shape of this molecule. So the enzyme that's actually going to split it in half will be able to recognize it and work on it. So in this form, in the beginning, it's not something that the enzyme that will end up splitting it in half can actually work on. It cannot be in this form. So the first reaction requires ATP. And what happens is that we have a phosphate group, a phosphate group, a phosphate group. This is it. This is your phosphate group right here. A phosphate group that's added to the glucose. So now it's called glucose 6-phosphate. And then you have another enzyme called phosphoglucose isomerase, which means that this that this molecule is going to change into an isomer of it. What does that mean? That means all of the atoms are still there. They're all the same on each carbon. There's the same number of oxygen and hydrogen. But what's different is, can you see a difference between these two? This is where you can pause and then think about it for yourself for a little bit before I show you. And I'm going to show you. You see the OH is up here? It's down here in this one. The OH was down here, and then now it's OH is at the top in this one. You see, just a tiny little difference, just how the hydroxyl group is pointing down, kind of below the molecule, if you're looking at it in 3D, and then the other one is pointing up in the molecule, right? If there's a plane, there's one of the hydroxyl group pointing down and the other one hydroxyl group pointing up. And they're pointing in opposite direction in the other molecule. Same number of carbons and hydrogens and oxygen and phosphate. That's why we call them isomers, because there's a different spatial arrangement of these functional groups. Okay. And then you have a phosphofructokinase. So there's an addition of another phosphate. And then finally, it's only in this form, fructose 1,6-biphosphate, because now there are two phosphate groups on this molecule that now we're calling fructose. And that's the molecule that the enzyme that can split it recognizes. So triose, phosphate, isomerase, none of these enzyme names you have to remember, but you just kind of have to get a feel for, okay, we're going through many different stages of that glucose, which becomes a fructose that's doubly phosphated instead of just one phosphate. And then it splits into glyceraldehyde 3-phosphate. This is very important. This you have to know. G3P. That's what it's called, G3P. Okay, and what do you have here? You have another G3P. Well, yes, because we have 6 carbons to begin with, and we're splitting them into two 3-carbon molecules. Look, 1, 2, 3 carbons, 4, 5, 6. These are the carbons. So if you numbered them in this molecule over here, these are the ones that are ending up in respectively in what I drew. So every time you start with a glucose, you're actually going to end up with 2 G3P. So the G3P then needs to continue. So the G3P is going to be transformed into pyruvate, and I don't think we need to go into the details of the phosphorylation and dephosphorylation and all of that. It just gets too complicated. So in the end, you have pyruvate. This is what we need to focus on. Pyruvate is the molecule that's at the end. 1, 2, 3. It's still a 3-carbon molecule, which means, oh, we must end up with 2 pyruvates. And you do. You end up with 2 pyruvates. Because for every single one of these G3P, we have this process going on. So the second G3P will see this process going on as well. Okay. Okay, so we saw that there are many enzymes that catalyze the reactions, um, and that the glucose is partially oxidized and broken down into two smaller sugars called pyruvate. So where did we write that here? That's here. That's it. Glucose partially oxidized and broken down into two smaller sugars, the pyruvate. See? Boom, 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 and here we go. Two G3Ps. So the net production is 2 ATP. Okay, we get 2 ATP molecules out of this little first step of glycolysis. We're good. And where is this ATP needed? It's actually needed in those reactions. The first reaction over here and the third reaction over here. And where is ATP being produced? Right here. When we go from phosphoenol pyruvate, we have an ATP produced. And the other ATP needed is your 1,3 biophosphoglycerate. You need, you have ATP production when that chemical reaction takes place. Okay, so we have 2 ATP that are being produced for every G3P over here. So this is a G3P. So for every G3P, we have 2 ATP molecules and 2 pyruvates. So we actually made 4 ATP, right? We made these 2, but we have 2 G3Ps. So in the end, we made 4. I just want to show you where the 4 is coming from. But because we used 2 ATPs in the beginning, really in the end, the net production of ATP was really only 2. So the phosphorylation of ATP takes place in an enzyme that is basically made for transferring a phosphate group from any substrate to ADP and regenerating the ATP molecule from that. Okay, so we talked about how glycolysis requires many enzymes to catalyze reactions. They partially oxidized and broken down into 2 smaller sugars. That's the second step. Well, that's the big picture, I should say, for glycolysis. The net yield of ATP is 2. And the last thing we need to talk about is the production of 2 NADH H+ molecules that will be used for later generations of more ATP. So here are the steps again, if you were interested in seeing it again. So where are those NADH H+ produced? Let's see. They are where? How come it's not written here? Yeah, so it should be, oh, it's here. You're probably looking at me like it's right there, Dr. Jabor, it's right there. Yes, so I still had this picture in mind over here, and we were looking at the ATPs. And I missed this one. So here we go. So NADH is created right off the bat after the G3P is appears. So this is your G3P. There's another G3P. They both 3 carbon molecules. come from the carbon, from the fructose, this intermediary molecule. So 2 ATP molecules are used up at the beginning of glucose. And in the end, we had 4 ATP molecules that were formed. And we had 2 NADH, 2H plus formed as well. So that's important. And the product of all this is pyruvate. We transformed this larger molecule into 2 smaller 3-carbon molecules. So there's an initial energy investment during glycolysis, and so sometimes it pays off if we actually use the energy in the cell, and some other times it doesn't pay off because the cell is really not very active. And here are your electron acceptors. NAD plus is going to come down to NADH. And the pyruvate is the molecule that's going to go to the next part of the story. So the net use here of all of this is very important. Glucose turns into pyruvate, 2-pyruvate, and then we have ATP and ADP formed in the process. So here's another kind of summary. All right, so let's see what happens to this pyruvate. The pyruvate that was generated from glycolysis, right? We're done with stage 1. Glycolysis is over. We end up with 2 pyruvate molecules. So pyruvate is going to get oxidized into what we call acetyl-CoA, and then completely to carbon dioxide via the citric acid cycle. Because remember, the last product of all these carbons, it's going to end up being carbon dioxide gas. So pyruvate, that 3-carbon molecule, really eventually is going to end up being carbon dioxide. But first, it's called acetyl-CoA, and then it goes through what we call the citric acid cycle to become carbon dioxide. Okay. So pyruvate is reduced to lactate via lactic acid fermentation if there is no oxygen available. So there is a fermentation process that takes place, and it actually turns into lactate. And lactate, which is also lactic acid, is it's acidic, so it actually burns, and that's what accumulates in your muscles if you're like exercising and you're not breathing fast enough, and your cells are basically not getting enough oxygen on time. They're taking this glucose and making pyruvate and transforming pyruvate into lactate instead of carbon dioxide and exhaling it. So you end up with lactate in your muscles, and that's basically what gives you that muscle soreness, the acidity of the lactate. Okay, and there's another fate for pyruvate in the absence of oxygen. So this is with oxygen, and this is without oxygen. In the absence of oxygen, pyruvate can also be converted to ethanol, that's alcohol, and carbon dioxide, and that's called alcohol fermentation. Well, our body doesn't do that, but it happens in plants, and where you end up producing ethanol out of the sugar in the plant. Our body can do lactic acid fermentation, and that's what happens when we don't breathe enough oxygen when we're exercising. No, don't breathe enough oxygen and exhale, and we don't exhale CO2 because in the process we're making lactate. But so I'm showing you that there are three outcomes possible for pyruvate. We're going to focus on the normal outcome of pyruvate as normal cellular respiration is taking place. Okay. So here's the diagram that goes with the previous slide. From glucose, we have, after a series of chemical reactions, we end up with two pyruvates, right? This is one 6-carbon molecule. The two pyruvates are two 3-carbon molecule each. All right. If we have oxygen, so under aerobic conditions, we will end up with acetyl-CoA. So each one of those pyruvates will give one acetyl-CoA. And then through the cycle, citric acid cycle, we will end up with carbon dioxide exhaled and some water molecules. So this happens in animal, plants, and many microbial cells under aerobic conditions. Now, if we don't have enough oxygen, we end up going through production of lactate. So this is under hypoxic, which means low oxygen levels, or complete anaerobic conditions, which means no oxygen levels. So hypoxic, for example, when you're not breathing enough oxygen. But anaerobic means no oxygen at all. You end up with lactate, fermentation to lactate, and so this happens in muscles, and erythrocytes are your red blood cells, and some other cells in some microorganisms. So that's one fate. And then another fate that only takes place in yeast. I should have said yeast, not plant, but yeast. Yeast is the one that is actually producing the ethanol, using the sugar in the plants. The two pyruvate turn into two ethanol molecules. All right, but we're going to focus on the normal aerobic conditions, pyruvate oxidation, followed by the citric acid cycle. So again, during glycolysis, you have two pyruvate that are made in the cytoplasm of the cell. Remember, you need to know where these are taking place. Pyruvate must enter the mitochondrion to participate in the citric acid cycle. The citric acid cycle is inside the mitochondrion. So this portion here, the citric acid cycle, is inside the mitochondrion. So we have to get in there to do this stage 2. Okay. So during pyruvate oxidation, the pyruvate is actively transported across the outer mitochondrial membrane and converted to acetyl-CoA. So let's see where this is happening. There we go. It is outer membrane right here. Okay, so it goes through the outer membrane, freely permeable to small molecules and ions. And then in the inner membrane, it's impermeable to most small molecules and ions. And this is where we're going to have, the machinery for the final third stage, the third and final stage of cellular respiration. So we'll talk about these electron carriers when we get to that. In the matrix, we have pyruvate dehydrogenase complex, the citric acid cycle. This is where it's going to take place. So it's going to take place floating here in the matrix. And we have, many other things. You don't have to memorize all of these details. So here's where pyruvate oxidation takes place. Right here. There's a transport protein in the inner membrane. Pyruvate is right here. 1, 2, 3 carbons. And I want you to follow these carbons. One of these carbons is going to turn into, and you all can already see it. Look at this. This is your CO2 gas. So pyruvate first has to go in. Here we go. It goes in, and in the process of going in, it loses that CO2. Okay? And you're left with a small 2-carbon molecule. The small 2-carbon molecule grabs a complex called coenzyme A. It just takes it in. And this small 2-carbon molecule, after it grabbed the CoA complex, so the enzyme, coenzyme A, is bringing that CoA complex, and it attaches it to pyruvate, which lost the carbon dioxide to begin with. So here the smaller 2-carbon molecule grabs on the CoA and becomes acetyl-CoA. So this is what you have to remember. This is important. Pyruvate enters in the inner membrane, it loses a CO2, and at the same time, it's going to grab a CoA and become acetyl-CoA. To do that, you also need NAD+, because you're going to break bonds here. You're breaking a bond to release CO2, and you're going to create a bond here. This is a bond that's created between CoA and this very small molecule. So as you're breaking these bonds, you're going to capture these electrons that are from these bonds into NAD+. NAD+ becomes reduced into NADH, and that basically allows you to take those electrons out of there. And coenzyme A is added on top. So NAD+ is going to get reduced into NADH, and yeah. And so this is one more NADH that's being made. Remember, we produced 2 NADH during glycolysis. I just want to remind you. Here we go, 2 NADH during glycolysis. We produced a net number of 2 ATP. We actually produced 4, but we also used 2. So we really, in the end, we only produced 2. And we produced 2 NADH. But look at this. For every pyruvate, we have an NADH made. So for every 2 pyruvates, because that's what we have at the end of glycolysis, is 2 pyruvates. Remember, 1 6-carbon molecule gives off 2 3-carbon molecules. So really, in the end, for every pyruvate, we get an NADH. So really, we have 2 NADH made. So we can do here net reaction pyruvate oxidation is this. And we can say times 2 times 2 times 2 times 2. All right? Because glycolysis released 2 pyruvates. Okay, now we can enter the citric acid cycle, and that's a lot of chemical reactions there, but it's going to be reduced to the big picture. So acetyl-CoA, now we have that. This is what we have now. Acetyl-CoA enters the citric acid cycle, and that's where it will be completely oxidized to carbon dioxide. Because remember, every single one of these, here we go, every single one of these carbons will eventually be carbon dioxide. So even though we made a couple here, it's not enough. We still have, these 2, right? We got rid of this one during pyruvate oxidation, but we still have these 2 that we need to also turn into carbon dioxide. So in the process of turning those remaining carbons into carbon dioxide, so going through the citric acid cycle, in that process, we actually end up making one more ATP, three more NADH, and one FADH2, because that's the first time we see FADH2 in the story. Wow, one more ATP. Look at that. We already made two. We already made two during glycolysis. We made two more during pyruvate oxidation. Right here, right? Two more during, oops, no, sorry, that's the glycolysis. No, there's no ATP during pyruvate oxidation. Sorry about that. So two during glycolysis, and one more during the citric acid cycle. Okay, so three. So it's not such a big deal. And actually, the most of ATP is going to be made all the way at the end. So here's your cycle. Here's your cycle. For every one acetyl-CoA, we're going to go through all of this craziness. And the idea is to produce NADH, and FADH2. That's very important. Yes, in the process, we're also producing an ATP here. And another NADH. In the process, we'll produce one ATP here. Great, but it's not that much. Remember, we're producing 30 all the way at the end. So this is kind of bonus, but it's really not enough. So the citric acid cycle will produce one ATP, three NADH, and one FADH2 for every one acetyl-CoA. Remember, we have two acetyl-CoA to deal with. So all this doubles up. So for every acetyl-CoA that comes into the cycle, that's what comes out of the cycle. So if you want to, you can kind of follow this color coding of these carbons as they're going through here. And that way you feel for, okay, the new carbons coming in, they get added on to, I'm trying to like avoid using the names of all of these, intermediate molecules. But they're getting added on to this 4-carbon molecule. So in the cycle, we have a 4-carbon molecule, right? 1, 2, 3, 4, yes. And it becomes a 6-carbon molecule. And the 6-carbon molecule is actually going to be split into a where is it happening? Now I lost track of this. CO2, CO2, CO2. Okay, so you can follow the new ones coming in. This is what I meant to say. The new ones coming in are in red, and they actually stay in the molecule. And the ones that used to be in the previous molecule are the ones that are going to be exhaled as CO2. So it basically takes a whole cycle for the carbon to actually be transformed into carbon dioxide. Okay, and if you don't remember that, that's okay. The idea is acetyl-CoA goes in, carbon dioxide is exhaled, because that's the goal. And that's one of the products of the citric acid cycle. The goal is to make NADH, 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 and FADH2. That we have an ATP molecule made too, that's bonus. But these are the ones, NADH and FADH2, that are going to be needed in the next and last phase, the last stage, at the inner membrane of the mitochondrion. That's where the biggest amount of ATP is going to be made. That's the most important part back there. And we need NADH for that story to take place. So these little guys here are going to be very important for the next stage. So here for us, we're just going to reduce it to what is the net reaction of the citric acid cycle. For every one acetyl-CoA going in, we have two 2 carbon dioxide going out, even though technically speaking, those 2 carbons are not, the ones that are coming out in the first round. It actually takes a second round to make them go out because it's the carbons from the previous round. You see the blue here? Okay. But overall, the 2 carbons that come in, eventually they come out. That's it. We get rid of them. Remember, all the entire 6 carbons glucose molecule is going to be 6 1 carbon CO2. Okay. Okay, 1 ATP, that's bonus, but the most important is 3 NADH and 1 FADH2 for every 1 acetyl-CoA coming in. All right. Finally, let's get to the last stage, oxidative phosphorylation. And I might do a separate video and literally draw this out. Even though I have diagrams here, I think drawing it out and taking the time to just draw it out, it just allows you to kind of absorb and digest everything that's being said and just see it and do it for yourself. So if this video is not available yet, create your own drawing. Okay? Redraw this for yourself. All right, so let's talk about oxidative phosphorylation. So this is the last stage, and this is when we actually producing that famous ATP. Because remember, that's what we want. We want energy production, that glucose that we ate from that pink cake back then. That glucose, the purpose of it is to make ATP, the energy for the cell. So the production of ATP is going to be derived from the redox reactions of the electron transport chain. And how do we get that electron transport chain to work? Is we need the energy from NADH and FADH2 to be dropped off, right? They're coming from the citric acid cycle, and then they come in here and they say, hey, here we are. We just came from the matrix where the citric acid cycle was spinning over there. And here we are, we just got made, we're fresh, and we come here to the inner membrane of the mitochondrion, and we're like, use us, take our electrons, and let's go. That's what they're here for. They're here to bring their electrons, dump them onto this electron chain so they can be used up for what I'm going to tell you in a minute. Okay. So here's the electron transport chain. So it's a series of membrane proteins. Here they are. 1, 2, 3, 4. It's a complex of 4. 1, 2, 3, 4. Yeah, let's just say that. 1, 2, 3, 4. Here they are. And then there is a we don't really even need to talk about those. A Q complex and a cytochrome C complex over here. The electrons. So let's start the story there. Here are the electrons. This is NADH. Okay, NADH. That NADH came from the citric acid cycle. This is where it came from. That citric acid cycle that's kind of like, spitting out these NADHs. NADH, remember 3 NADH and 1 FADH2, right? So this is where these guys are coming from. This is coming from there, and this FADH2 as well. Okay, so this NADH comes in to dump its electrons in here. This complex at the membrane, these blue proteins sitting in the membrane, in the inner membrane of the mitochondrion, they don't move. These blue proteins, they're there. And their job is to grab these electrons from NADH and transfer it to the next one. And that energy from taking these electrons from NADH, that energy from these electrons drives the movement of hydrogen ions through the membrane. That's the purpose of these electron transporters, NADH and FADH2. When they come to the membrane, they're dumping their electrons. They're going to get sucked up by these electron transporters in the chain, this electron transport chain, those complex blue proteins there. And that's going to allow these transporters here to pump hydrogen ions against their gradients. So these are hydrogen ions that are getting accumulated in this intermembrane space between the two membranes of a mitochondrion, this intermembrane space. And we are accumulating them. The concentration is getting bigger and bigger and bigger, which means every time we're putting more in there, we're going against their gradient. That requires energy. We're basically shoving them in that space. Remember, the mitochondrion is a double membrane. I'm going to draw it like in red here, just so you can see. There's an outer membrane and an inner membrane that's so big it folds on itself, right? Because it's so big. So this is the inner membrane. Oops. This is the inner membrane, and this is the outer membrane. We're talking about all of these are in the inner membrane. They're here. Here, I can even do them in blue so you can see that. These are the proteins that I'm talking about, these proteins over here. And what are they doing? They're pumping hydrogen ions in that space. How are they pumping these hydrogen ions? With what energy? With the energy released from the electrons that are being plucked off NADH. So when you oxidize NADH, you take energy out of that molecule, and you're basically using that energy to pump the hydrogen ions against their gradient into this small space here between the two membranes. We're talking about this space here. And they're accumulating and accumulating. Okay, so that electron is going to be transferred, right? So that electron, the movement of that electron through here provides that energy for the pumping of hydrogen ions, but it's transferred to the next set of proteins, the complex 2, that has a Q complex with it. So this is complex 1, complex 2, 3, and 4. Complex 2 also has electrons that are coming from FADH2. So it's able to take on these electrons and the electrons from the previous complex and pass both on to complex 3. And complex 3 uses that energy to pump more hydrogen ions. And complex 4 pumps additional hydrogen ions. So that's how come you have so many hydrogen ions in that space, because it gets pumped thanks to the energy that's kind of traveling through these complexes. So these are electrons that are going from one space to another. Here, let's put it in yellow so we can see it better. Here, electron over here, electron over here. So literally these electrons are like being here. It's like a hot potato. It's like that's an American expression for non-American viewers. It means, hot potato is something like you don't want to hold it because it's too hot, and you just can pass it on. It's even a game in America. So, it's like these electrons, like, oh, I got them, great, that's energy. Now I don't need them anymore. And they're passed on to the next complex, and the next complex says, oh, what do I do with this? And the last complex gives these electrons to oxygen. Oxygen gets reduced into water. So when oxygen grabs on these electrons, it gets reduced into a water molecule. But the passage of these electrons through complex 4 was enough to also provide that energy for the pumping of hydrogen ions into that intermembrane space. So what happens next? We have chemiosmosis. So what the heck is that? It's the last piece of the story. After you've pumped all of these hydrogens, oops, let's not do yellow because we were doing blue. All of these hydrogen ions in that intermembrane space, right? This is the intermembrane space over here, right? Here, the electron transport chain, we're only showing one complex, and it's like just random. This represents all the complexes, right? Oops, right? Those four. Basically, we're talking about these three out of the four are pumping, pumping, pumping. So this is representing the whole chain, pumping hydrogen ions into that space. Okay, so what happens next? You have all of these hydrogen ions that are packed in here. And here you have a an ATP synthase. It's a membrane protein that rotates, and in the process of rotating, it's going to produce an ATP molecule. What? This is so cool. So the ATP synthase rotates, and in the process, we'll take an ADP plus a phosphate group, click them together, and form ATP. Oh my gosh. Okay. So what energy is it going to use to rotate? Things don't just kind of like spin by themselves. It uses the energy that's going to come down from the rush of hydrogen ions down their concentration gradient through that synthase. Because when that door is open, it basically allows all of these hydrogen ions to see a compartment where they're less concentrated, the one on the other side. And it sees through that hole, because there's a hole there, it's like a tunnel. It sees through the hole, oh my gosh, the compartment on the other side, there's much, much, much less of us. Let's go, because we're too crowded in here. And solutes and ions and molecules, they don't like to be crowded. If they have a chance, if there's a door open or, a channel, if there is a way of getting to the other side, they're going to take it. And that's going to happen naturally because that's diffusion. That's a passive process that doesn't require energy. But when you accumulate them in such amount, their passage through the tunnel actually generates the energy and energy. And that energy is harvested by the synthase itself and uses that energy to spin. The energy coming down from the rush of the hydrogen ions coming down spins that ATP synthase, and it produces an ATP molecule. And we have so much of these hydrogen ions rushing down this ATP synthase that we make 28 to 30 of these molecules in this process. So it's very, very productive, much more than the two made during glycolysis, and during citric acid cycle we have one more. This is like boom, we're making so many more ATP molecules. Okay, so this is a little bit explanation about how these complexes work. It's a series of electron carriers. Each one is an electron carrier. They're basically accepting the electron from the NADH, which is also an electron carrier. So that NADH is bringing, right? It's bringing the electrons to the complex. The complex is accepting the electron, and then it's going to pass it on to the next complex. So that's why we call it a transport chain, an electron transport chain. They're literally sharing the electrons from one to the next. Hey, look what I got. Take it, take it, take it. It's too hot. So the electrons are passed between electron carriers, and as electrons pass or are passed, energy is released, and the energy released is what's used to pump and pump and pump these hydrogens against their gradients. And they are pumped in that intermembrane space. Okay? Okay. And after they accumulate here and they, we've reached, these electrons reach the end over here, this ATP synthase opens up and you have a big rush of hydrogen ions down this tube, and it allows the synthase to spin, and that spinning allows for the reaction of ADP plus phosphate to create an ATP. Isn't it amazing? Okay. And we reminding you, we also need to, what do we do with these last two electrons? Well, we give them to oxygen, and in the process, we end up making a water molecule. So the last electron acceptor in the chain is oxygen, and water is produced in the process right here. Okay, so for 2 electrons, this is what we would have. So of course, for every oxygen molecule, we actually have double that. So NADH plus and FADH2, those were made in glycolysis and the pyruvate oxidation and the citric cycle. They were made a little bit here, a little bit here, a little bit here. All of them go to the electron transport chain. That's very important because it's their energy. They're going to release electrons there, and that release provides the energy that's needed to pump hydrogen ions against their gradient. So NADH and FADH2 contribute their electrons to the first, to the electron carriers. So NADH to the first one, FADH to the second one. This is what we saw here and here. And then finally, the concentration of hydrogen ions being so high on one side, when you open the ATP synthase tunnel, the hydrogen ions will flow down their concentration gradient, That's a passive transport. So that movement itself doesn't require energy because it's from high to low, right? From high concentration to low concentration. But so it doesn't require energy, but the movement of these hydrogen ions down this tunnel, it literally generates the energy necessary for the ATP synthase to spin. So hydrogen ions flow through the protein. ATP synthase catalytic site is activated, right? Because it has this enzyme, the catalytic enzyme that will allow for the production of ATP. And ATP is made from ADP and a phosphate group. And here's the ATP synthase, if you're interested in another view of it. It's just amazing that, we know so much now about that. There was a time where we thought maybe it was just one big blob in the membrane. Nobody had any idea of what the structure of ATP synthase was. And, it was probably 1 to, I don't know, 10 dozen graduate students that worked on these research projects to characterize, okay, this molecule, what is it made of? And then they discovered, oh my goodness, there's different parts to it. And what do these parts mean? What do these parts mean? And how are they all put together? And it's just, the amazing discovery of every single thing that makes up these cells. So whatever we draw, like just one round circle in the membrane, really in reality it still has yet to be discovered of what it's made of. So certain things we do know how they're structured because they were studied and discoveries were made, and the information was known afterwards. So for every NADH that contributes electrons to the electron transport chain, 3 ATP molecules are generated. For every FADH2 that contributes electrons to the electron transport chain, 2 ATP molecules are generated. I forgot why this one is back here. Oh, just to show you the difference with the, I forgot about that. To show you the difference with the number of ATP made uh, in the other processes in the fermentation. We don't have ATP made as much in fermentation, and that's a problem because otherwise we would be able to survive on fermentation, but we can't. We need ATP, that's this energy for the cell. That's why we need oxygen, because if we don't have oxygen, we're not going to be able to make enough ATP molecules, and the cells are going to die. Okay, so I already talked about fermentation here. Maybe I should have moved these two slides a little bit sooner. So ancient prokaryotes are thought to have used glycolysis long before there was oxygen in the atmosphere, and that's kind of where fermentation, originally probably came from. Very, very little oxygen was available in the atmosphere until about 2.7 billion years ago. So early prokaryotes likely used only glycolysis to generate ATP at the time, and glycolysis, because oxygen is needed at the end. So glycolysis makes ATP, and those two ATPs were enough for, those very primitive cells billions of years ago. So it's a very, very ancient process, glycolysis, that cells, used to make even before they were able to use oxygen. So proteins, carbohydrates, and fats, they all eventually will go through glycolysis. So we do produce energy from all of these food sources. We use the example of glucose because it's straightforward, and this is how glycolysis takes place, is with glucose. But fats contribute to this glycolysis by skipping the first step here and providing this intermediary molecule called glyceraldehyde 3-phosphate from glycerol, the glycerol that comes from the triglycerides. There's a glycerol in fatty acids, and you learned that in a previous chapter that we covered. And that glycerol turns into glyceraldehyde 3-phosphate, and now we can kind of go into the whole process of making ATP using fats. So even if we don't have sugars, we can still use the fats and make ATP. We can use proteins and make ATP. Proteins will contribute at the level of pyruvate. Their carbons here are rearranged to make pyruvate, and once we have pyruvate, we can do the rest of the story and produce ATP. So regardless of the food we eat, we always can generate energy. That's the point of this slide. So this is just showing you at which stages we can contribute to this whole story, whether we are a protein, a carbohydrate, or fat. And I don't typically ask about that on an exam per se for, in which exact stage are proteins involved in ATP production, right? Obviously, they don't start at the level of glucose because they're proteins. And unlike fats, they don't produce glyceraldehyde 3-phosphate. So, but they can produce pyruvate. So if they produce pyruvate, perfect. Then we can go into that whole story, but at the pyruvate level. So I don't ask at which level these go into, but I do expect you to know that whether it's carbohydrates that generate the glucose immediately, or it's proteins or fats that jump into the story a little bit later, regardless, we have all of these stages. They can take place for ATP production. Okay, that's it. That's the end of the lecture. We made ATP. I wanted to see if I had missed something. I think I just, yeah, I think I just covered everything. I did. I covered everything, and I'm going to try to do an actual drawing of this because I think it just allows to really see what's going on and tell a story with a drawing. I'll see if I can record that and post it. But regardless, try to do your own drawings. And again, learn it stage by stage. Otherwise, it's too overwhelming. There's too much going on. Just understand the big picture and then take every stage at a time. So one night, learn about glycolysis. What the heck is glycolysis? Where does it take place? We start with this, we end up with this. Okay, I learned glycolysis. The next night, you learn what happens to pyruvate, which is the result of glycolysis. That pyruvate, what happens to it next? So then learn about that and those and that part of the story. And then, on a third night, you can, go study what's going on at the membrane of the mitochondria and that oxidative phosphorylation. What the heck is that? What's going on there? And learn that. Because, doing everything in one night is just too much. And I also recommend watching animations, amazing animations that show you the ATP synthase spinning, and also animations that basically tell the whole story from start to finish with, movement of all these ions. That way you can visualize what's going on. I have them in my in a playlist in my YouTube channel. I don't know how to link that to this particular lecture, but you know how to go to my YouTube channel and find the playlist and watch these animations for cellular respiration. So it's probably under cell biology playlist. All right. All done. Thank you for paying attention.